Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
It's, it's quite sobering, isn't it? To think back to the early days of the pandemic. The narrative was almost, well, reassuring about infants and COVID-19. They seemed relatively okay.
Yeah, that was the initial impression.
But the more recent information, it's painted a, frankly, more concerning picture. Significant numbers of babies, really young ones under 6 months, needing hospital care.
Exactly. And that immediately makes you ask, well, what is actually happening? How is this virus interacting with their immune systems, which are, still figuring things out?
Right. That's the core question.
And that's precisely what we're digging into today. We're doing a deep dive into this really fascinating new scientific study. It looked meticulously at the immune responses in infants who were hospitalized with SARS-CoV-2 across different levels of severity.
And this isn't just a quick overview. We've got the nitty-gritty from a detailed scientific paper.
Yeah.
They use some pretty advanced techniques, like looking at the gene activity in single-cell ribonucleic acid sequencing.
Yeah, and detailed blood analysis, too.
Plus, we're bringing in context from a health news article that sort of tracks this changing understanding of COVID in the littlest patients.
Right. So our goal here is really to understand the key ways these very young immune systems react to the virus. How does it change if the illness is mild, moderate, or severe? And importantly, how does it stack up against what happens in adults?
We'll try to cut through the scientific complexity.
And bring you the most important takeaway.
Okay, let's get into the study itself then. What were the researchers aiming for, and how did they set it up?
So the main goal was to really thoroughly map out how these young infant immune systems respond when infected with SARS-CoV-2.
Okay.
To do that, they collected and analyzed blood samples from 26 infants. These were all babies hospitalized with the virus, and they were grouped based on how sick they were, subacute, moderate, and severe.
And they needed a comparison group.
Yes,
Yeah.
exactly. They also included 14 healthy infants as controls, and we're talking really young babies here. The median age for the infected infants was about 1.6 months, so around 6 weeks old.
Wow, tiny.
Yeah, and the healthy controls were around 2 months old on average.
So what kind of tests did they run on these blood samples? What methods were they using?
They used a few pretty sophisticated methods. A really key one was that single-cell ribonucleic acid sequencing. They did this on peripheral blood mononuclear cells, PBMCs, basically.
Okay, what are those exactly?
Think of them as the main immune cells circulating in your blood. T cells, B cells, monocytes, that whole mix.
Got it.
And doing it at the single cell level lets you see exactly which genes are switched on or off in each individual cell. Gives you an incredibly detailed picture.
Much more than just looking at the blood overall.
Precisely. They also measured levels of 92 different inflammatory proteins, cytokines in the serum, the liquid part of the blood.
92. That's comprehensive.
Very. And they looked at antibodies, specifically antibodies against SARS-CoV-2, but also against the common cold coronaviruses. Plus, they measured the viral load, how much virus was actually present in the infants.
Okay, so a multi-pronged approach. When they looked at the types of immune cells, what were the big changes they saw in the babies with COVID?
Well, they definitely saw significant shifts in the proportions of different immune cell types. But maybe the most striking finding was this widespread interferon-stimulated gene high state.
Interferon-stimulated genes. So genes triggered by interferon.
Exactly. Interferon is like that first alarm signal the body sends out against viruses. Seeing high activity of genes stimulated by interferon tells you there's a really strong early antiviral response happening.
And this is across many cell types.
Yes, quite broadly. It included CD14 positive monocytes, those are key scavenger cells. Also naive CD4 positive T cells, the helper T cells that are new to the game, and even proliferating cytotoxic CD8 positive T cells, killer T cells that were actively multiplying, plus naive and transitional B cells, the antibody factories in training.
Wow, so a really system-wide interferon alert, it sounds like. Did it differ depending on how sick the infants were?
Yes, that's where it gets more nuanced. Infants with moderate and severe disease had more of those CD14 positive monocytes. And interestingly, these specific monocytes were showing high gene activity related to both interferon and inflammation simultaneously.
Both antiviral and inflammatory signals firing strong in the same cells.
Right, which suggests, a particularly intense early reaction in the sicker infants.
Now, you mentioned monocytes. I remember hearing about CD16 positive monocytes being affected in adults with severe COVID. What about in these infants?
That's a great point, and it highlights a key difference. In adults with severe disease, those CD16 positive monocytes often get depleted, their numbers go down.
Okay.
But in these infants with moderate to severe COVID, their levels stayed normal. And the reason was an increase specifically in CD16 positive monocytes that were also in that high interferon stimulated gene state.
So maintained numbers, but highly activated by interferon, another distinction from adults.
Exactly.
What about dendritic cells? They're important for kicking off the adaptive immune response.
Yeah, they are. And infants with moderate to severe disease actually had fewer conventional dendritic cells. Plasmacytoid dendritic cells, which are big interferon producers themselves, were also lower in the severe cases.
Lower numbers, but were they still active?
Interestingly, yes. Within that reduced population of plasmacytoid dendritic cells, they found a subset that showed high interferon-stimulated gene expression. So even though there were fewer, some were still strongly contributing to that interferon signal.
Okay. And they also mentioned megakaryocytes. What's their role?
Those are the cells that produce platelets involved in blood clotting. They were increased in the infants with subacute and severe illness.
Okay, let's shift to the T cells, the soldiers of the immune system. What was happening there?
Well, similar to other cells, naive CD4 positive T cells, the helper cells showed higher interferon gene activity, especially in moderate and severe cases.
So they were getting that interferon signal loud and clear.
Definitely. For memory CD4 T cells, the ones that remember past infections, it was a bit mixed. Some subsets had increased interferon gene activity, but other memory subsets were actually reduced in number.
Okay. And the CD8 positive T cells, the killer cells.
They also showed increased interferon gene activity in moderate and severe cases. But here's something really striking, and another contrast with adults.
What's that?
They saw an increase in CD8 positive T cells that were actively proliferating, multiplying, and had a cytotoxic profile, meaning they were geared up to kill infected cells.
And that's not typical in severely ill adults.
Generally, no. That profile of actively multiplying killer T cells isn't a hallmark of severe adult COVID-19 in the same way. It suggests a potentially different dynamic in the infant cellular response.
That is a significant difference. What about the B cells, the antibody producers?
A similar theme there. Both the naive B cells and the transitional B cells, sort of teenage B cells, showed high interferon gene activity in the moderate and severe groups.
So the interferon signal reached them too.
Yes. And they also spotted a small group of memory B cells that had markers suggesting they were on their way to becoming plasma cells, the real antibody-secreting factories.
Okay, and zooming out from the cells to the blood plasma, what about those inflammatory proteins, the cytokines?
The infected infants, especially the severe group, had higher levels of various inflammatory cytokines and also interferon gamma in their blood compared to the healthy babies.
So more inflammation circulating in the sicker infants.
Correct. Indicating a more intense overall inflammatory state.
And despite all this focus on the early interferon response, did the infants actually manage to make their own antibodies against SARS-CoV-2?
Yes, absolutely. That was clear. They consistently developed immunoglobulin G antibodies, IgG, a standard type of antibody against different parts of the virus.
That's good news.
It is. And importantly, they checked for autoantibodies against interferon antibodies, mistakenly attacking the body's own interferon signal, which has been seen in some severe adult cases. They didn't find those in these infants.
Also good. And did the antibodies they might have gotten from mom against common colds help at all?
It seems not. They noted the presence of antibodies against seasonal coronaviruses, likely maternal, but these didn't appear to prevent the infants from getting infected with SARS-CoV-2.
Right. So bringing it all together, how did this whole immune picture in infants compare overall to what we know about adults?
Well, the interferon-stimulated gene signature in monocytes was somewhat similar, but the really big difference was in the lymphocytes, the T cells, and B cells.
How so?
In infants, especially the severely ill ones, that interferon signature was much, much stronger and more widespread in T and B cells. It was like almost all their lymphocytes flipped into this high interferon state.
Whereas in adults?
In severely ill adults, typically only a fraction, a minority of T and B cells show that same intense interferon activation. So it's a far more dominant response in the infant's lymphocytes.
Okay, that's a lot of detailed findings. What are the main implications then? What does this study really tell us?
I think the first big takeaway is that infants, even just weeks old, can mount a really robust immune response to SARS-CoV-2. It's not weak or absent.
But it is distinct.
Yes, it's definitely distinct from the adult response in several key ways we've discussed.
And that really strong, widespread interferon response seems central. Why might that be so important for babies?
Well, it strongly suggests that this interferon system is a critical, maybe the critical first line of defense for them. Their adaptive immune system, the T and B cells that learn specific targets, is still developing, still maturing.
So they lean heavily on this immediate, broad antiviral alert system.
That seems very likely, yes. It might be crucial while the more specific defenses are getting up to speed.
But you also mentioned that in severe cases, some cells had both interferon and inflammatory genes activated. Is that potentially a double-edged sword?
That's a really important question the study raises. That co-expression, especially in monocytes in moderate and severe cases, makes you wonder about the interplay. Is that combination helpful, or could it contribute to some of the problems seen in severe infant COVID-19? The balance might be different, or perhaps less finely tuned than in adults.
And the proliferating killer T cells in severely ill infants, that specific finding.
Yeah, that's really notable. Seeing those actively dividing cytotoxic CD8 T cells suggests a different kind of cellular attack pathway might be more prominent or operate differently in severely ill infants compared to adults. It warrants a lot more investigation.
It's also reassuring, though, that they do make antibodies.
Definitely. Even with what we consider a largely naive immune system, they clearly can develop humoral immunity, the antibody-based response to the virus.
Now, the researchers compare these findings to another study that looked at older infants who had milder disease. What did that comparison add?
It suggested that the really intense, widespread interferon signature they saw in these hospitalized infants might be specifically linked to disease severity. The older infants with milder illness didn't show quite the same extreme interferon profile.
Okay, that context is helpful. Now, every study has limitations. What were the main ones here?
Right, the researchers were upfront about that. One is the study size. It was modest, especially the severe group. Recruiting very young hospitalized infants for research is challenging, understandably. Yeah. Another point is that they mostly looked at the immune response at one snapshot in time during the hospital stay, apart from some antibody follow-up. So we don't see the full picture of how the response evolves day by day.
Like a movie versus a single photo.
Exactly. Also, the study focused on blood. It didn't measure the immune response directly in the airways, the nose and throat, which is where the infection starts. That mucosal immunity could be really important. And lastly, they noted a slight difference in enrollment timing for the subacute group compared to the others, which might have slightly influenced the interferon levels observed in that specific group.
Okay, so keeping those limitations in mind, what did the researchers themselves say about where we go from here? What are the bigger picture thoughts?
They really emphasize that this work highlights how much more we need to learn about the unique features of immunity in early life, especially facing viruses.
It's a specialized field.
It is. They suggest that while this strong infant interferon response is likely protective initially, we also need to explore if, in some situations, it might actually contribute to harmful inflammation or pathology.
The balance question again.
Right. They call for more longitudinal studies that follow infants over time, especially those infected with different common respiratory viruses, to really grasp the functional importance and long-term effects of these early life immune responses.
And that unique killer T cell finding.
They definitely flag that for more research. Understanding that specific population could give us really valuable insights into severe infant disease.
And ultimately, the goal is better care.
Yes, they believe that digging deeper into these unique infant responses could pave the way for more targeted ways to prevent or treat COVID-19 specifically in this vulnerable age group.
This has been really illuminating. It's clear that infant immune systems aren't just miniature adult systems. They have their own playbook for dealing with this virus.
Absolutely. This study gives us a fascinating glimpse into that playbook, but it also shows us how many pages are still left to read, you know?
Yeah, definitely. So as we wrap up this deep dive, what's a final thought or question you think is worth pondering?
Well, given everything we've discussed, this distinct, powerful, very interferon-heavy early immune response in tiny infants, it really makes you wonder, what are the long-term implications? How does experiencing this kind of intense immune activation so early in response to SARS-CoV-2 shape their immune system's development down the road? Does it affect how they respond to other infections later in childhood? This study cracks open the door to those really fundamental questions about early life immunity and its long shadow. It's a crucial area for future research.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.